Good morning. Good morning. Good morning. Good morning. Out of 19 lectures, this is the first I think that I've done that. So my apologies and thank you for letting me know that I was just speaking, flapping my gums. And I hope you can hear, and I hope the internet speed is working well. If it is not, my apologies for that. I can't really change it as we're going, but of course this is recorded, and so you'll have the opportunity to go back later on. Okay, I'm going to clear the letters that you've found. If you're interested in going back later on and finding these yourself, the solution's going to show. So cover your eyes, and let's talk about where we're going today. So last time, previously on Zoology 3700, we were thinking about, began our kind of chapter thinking about mobility and motion. We started out with kind of from first principles, like what are the pros and cons from an evolutionary perspective of being mobile or of being sessile. And then we spent the meat of that lecture talking about flight. And about, because we were talking about flight, we talked about the invertebrate lineage that is just the first to do it and the just kind of the best and the most abundant to do it. We talked about aerial plankton. We talked about great big beefy odonates and really tiny beetles and how the Reynolds equation and the viscous versus inertial forces that change as you become really, really small to really, really large affect how you fly. And so you can do it with like an Incredible Hulk kind of musculature of the odonate. 60+% of their total body mass is made up just of flight muscles, versus these feather beetles that are doing it with really super reduced wings that have really no membranes in between the veins. And in fact, on the veins, they also have further little hairs that allow them basically to claw their way up in the air column. But that's enough about last time. What are we talking about this time? We're talking about swimming and crawling, walking and running. And we'll move across the aquatic, marine, and terrestrial environment and across the Dun phylogeny as we deal with some of these examples. We'll talk about rotifers and ctenophores, squids and cnidarians, sea cucumbers and mollusks, onychophorans, echinoderms, and arthropods. Oh my. And my hope for you after this is that you can kind of interact with the scientific literature and with the biological world that in which you are a part to appraise, synthesize, and interpret examples about how invertebrates are moving, how they're swimming, how they're crawling, how they're walking, how they're running, how different environmental stresses like that Reynolds equation, how physical rules and physical criteria affect each one of those kind of mobility strategies, and in fact how their movement affects their ecological interactions. Okay, well, let's start. So we dealt with flying. Today we're going to deal with the rest: walking, running, crawling, and swimming. Now for swimming, which is where we'll start with a similar force diagram to what we talked about when we started talking about flying. And that was the fact that we have four forces that are affecting this squid that's trying to sit in the water column. That we have gravity pulling it down. That we have lift or buoyancy kind of lifting it up off of the substrate. That we have thrust, that's a force going backwards that's propelling it forward. And then we have drag that's dragging it down. And then inside of the biological world, as we get bigger and bigger, or as in this case, as you get smaller and smaller, drag increases. And this is, of course, this is the viscous, that kind of remembering back to our first 2 or 3 lectures together, thinking about the Reynolds number and about problems and challenges of being in the plankton. Because that Reynolds number, as a reminder, again, it's a dimensionless unit to think about the relative ratio of viscous versus inertial forces. And when it's less than 1, when it's much less than 1, viscosity is the thing. So for this rotifer, this is a viscous world. And when it's greater than 1, for a large squid, like a giant squid, then inertia dominates. And when we see lots of viscous forces dominating, we see a preponderance of ciliary or flagellar movements, like in this rotifer. But we see it across that kind of critical boundary. If you remember that diagram that we had that talked about the copepods as kind of sitting in that real transition point, a critical ecological, a food for so many things, but also as a transition in the biophysical world between less than one and greater than one, these forces kind of acting on them. But another group that does this, or that exists, or that brings with it the material or the movement strategies from the low Reynolds world, are the ctenophores. Ctenophores can exist very small. There are ctenophores, and particularly small ones, that exist early stages, larval stages, that exist in a low Reynolds number, which is, as I said, where we would see most of these flagellar or ciliary motions, the preponderance of that strategy being very successful. But there are some larger ctenophores that certainly exist in a greater than one Reynolds world, and they are still a group that moves through the world exclusively or nearly exclusively via ciliary action. And they seem to be the size limit of an organism that can be powered by cilia. Now, if you pause for a second, I'm going to sit on another train and draw your memory back to thinking about ctenophores in 2700. Ctenophores are different in a lot of different ways, and there's a whole bunch of evolutionary thinking, like the number of neurotransmitters they use, the fact that their anus is transient, and so they have a through gut, kind of, but not really. They have been successful, very successful, through hundreds of millions of years of evolution. They've largely done it in a different way. And this is kind of another way in which ctenophores have been successful in a different way. Most of the other eukaryotic animals never get bigger than a Reynolds 1 kind of world while moving by cilia, except for ctenophores. And so there's a whole bunch of interest in thinking about why and how. And we'll parcel that into our formation of ctenophores and put it on the side, and hopefully some of you will go off and become ctenophore biologists and then come back to this class in the future and tell us all about the things that you've learned. Now, I say that most of the taxa outside of this is my yes and about most taxa outside of ctenophores don't use cilia or flagella to move. Well, in fact, early life history stages of many taxa particularly marine ones, do use flagella and cilia to move because they use it in their early larval stages. So as they exist in the plankton, whether you're an echinoderm larva or a sea star larva or a snail larva or an annelid larva like you're seeing here, the surface area, the majority of that motion of movement is taken up by or contributed to by ciliated rings, ciliated bands, ciliated lobes. And that changes during development, particularly as they settle out of the plankton, if they're not a truly planktotrophic species. And it tends to be associated with feeding behavior at least as much as propulsion. And as they get bigger and bigger, it's less and less of the propulsion. Now, when we're thinking about marine taxa that move outside, the larger things that move without cilia, one of the immediate groups is kind of that taxon to the ctenophores, which is thinking about your cnidarians and medusoid cnidarians, where they use the jet propulsion. You remember you've got that kind of band constricting and the jelly kind of that is contracted upon, and then boosh, outside of the bell, as you see here, the water is expelled and the jelly moves itself forward. And so instead of like the ctenophore, which is swimming towards you and its ciliary tene rows are moving it towards you to feed on you, this is moving away and the mouth is facing kind of away from the direction of motion. But this is that propulsion, that expulsion of air. This is essentially jet propulsion that's interacting with the surrounding fluid that creates these stopping and starting vortices that during bell, the contraction of the bell and the relaxation. And the relaxing of that bell is kind of the rebound of the elastic mesoglia that brings it back into its original shape. So the top diagram, kind of frame from that last video that's moving through, this is the contraction phase, and this is the beginning of the relaxing phase here. So there's all sorts, in addition to cnidaria that move by jet propulsion, or the medusoids that move by jet propulsion, we see it a lot across some of your favorite invertebrate taxa, within the mollusks, the cephalopods, where of course we see kind of mantle edges moving you around, but then we see that you can fill up the mantle cavity, if you're a squid, or an octopus and quickly push out that water through the siphon. And the force of that water being ejected propels the animal in the opposite direction. So here, when you're looking at the vorticity or the propulsion forces of this squid moving forward, you can see some dense forces kind of right outside of the siphon that then kind of spiral off into the water. And this kind of, for whether you're a cnidarian or whether you're a squid, jet propulsion is great for escaping danger. It's maybe particularly if you're cephalized or if your feeding apparatus is opposite the direction of flow, it's not as great for chasing prey unless, as many of these mollusks can do, you can change the direction of the siphon so that you can then change it around and move forward. So we've got the squid that can do tail first swimming, where the siphon is pointing out and the water is propelled out, so water in, push, water out. We've got arms first swimming. This is an escape here. This is more predatory. So the siphon gets kind of elbowed around and directed behind, and that allows the animal to be jetting and propulsing jet-powered towards its prey item. In addition to this, we have the mantle. Remember the mollusk mantle that given time can become practically anything. So it's undulating, and it's kind of moving it through the water by a completely different method that we'll talk about in a second, but it has this as well. So it can do that and maintain its buoyancy or maintain its position in the water column as the mantle cavity is refilled until it contracts again to either jet away or towards its next prey item or away from its next predator. Now, squid are the exemplar taxon in our shared time together for us to think about the effect of larval of life history on your movement through a Reynolds continuum, a Reynolds world continuum. Because squid larvae, which you're watching one being born there, the paralarva, which is the squid biologist or the molluscan biologist word for the very youngest squid, they're very small. And the predominant physical forces that they face in the world are viscous ones because they're very, very small. However, if this is a lucky enough individual from these eggs to live, a giant squid could move through that Reynolds continuum until it's living in a Reynolds world that is like 10 to the 8th, so orders of magnitude into the inertial world. But the apparatus and the movement equipment that the paralarva to the giant squid have, so from the Reynolds below 1 and well above 1, doesn't really change. So how does that change? What does change as you go through the life history of this jet-propulsed animal? I'm going to clear my throat. And I hope that none of that came through. Well, the equipment doesn't change, but the morphology around the equipment changes. So one of the things that changes is that the paralarva, the very small things, have a much more rounded mantle. They're not streamlined. There's no dynamics to they're a blunt thing. Proportionally, they have much smaller arms. The funnel is much larger proportionally, and they have very rudimentary fins. So they're not using that kind of undulatory swimming motion that many of the adults do. So that's some of the key differences between the very young below Reynolds squid and the much larger, older, greater than high Reynolds number squid later on. The other difference is how they use, in addition to the morphological differences, is how they use that equipment. So this is actually, as the video is kind of looping through, this is an apparently bouncing paralarva. But this directionality is one of the strategies that they use. So the squid, the very tiny paralarva, predominantly use this vertically directed, high frequency, so that's the number of bounces, low velocity jet that allow them to bob up and down in the water column. So they're not using it so much to get away from a predator or to go towards a prey. They're just using it to maintain position. So they're like going they're like a little pipette that's just continually expelling that water to maintain their position and to hold position in their highly viscous world. And if you put one of them alongside all of its kin, you get this kind of gorgeous view that perhaps you've seen, the bouncing popcorn effect of the squid paralarva. So jet propulsion from cnidarians to mollusks, but also in our own neighborhood, or the neighborhood of the invertebrate world that is most adjacent to us. And that's, of course, in the Urochordates. So the thalassian, the pyrosomids, this is a Urochordate lineage that lives colonially. So this is a deuterostome. So we've jumped from outside of the bilateria to inside of the bilateria to inside of our own neighborhood. And to think about these pyrosomes, they live colonially. So each one of these, this kind of large tube that you're seeing the scuba divers swim around, is a tiny zooid that brackets a siphon, an opening, that brings water in, that is then pushed out by the whole colony out of this large opening. So that's what you're looking at right there. And this allows the entire colony to be jet propulsed. And this is kind of a departure from thinking about colonial animals as being sessile. Think like your coral and your bryozoan and that kind of an example. But here we have a colonial animal that's jet propulsed through the water. So jet propulsion widely distributed across the phylogenetic animal world. But as with the mantle of that squid, there's lots of other ways that you can move in the water. So for instance, you're looking at some videos of specialized sea cucumbers, so echinoderms that are using their kind of stubby feet to crawl across the ocean floor and to be deposited in filter feeders. But some of them can get up into the water column and begin to swim. And they develop thrust not by jet propulsion, but by undulating, as you're watching it there, or by flapping. So there's large undulations here, but then there are, especially in this next one, you can see there are kind of wings that allow the flapping motion to be continued, and the extension of that across and out into the water that help prevent it from floating immediately out of the water column. And we see flapping, we see it within the mollusks, within the mantle edge of the squids, we see flapping within the echinoderms, and we see flapping within other mollusks like the snails or the gastropods. So these are the snails, some of the Cleone, that have lost their shell and are moving through the water in using a highly adapted foot that's evolved into these wing-like paddles for swimming. And it uses a rowing motion to swim fast and to maneuver quickly. And so these are predators. These are predators of other sea snails, that some of which have still their shell. We talked about them in the filter feeding, where they have these large mucus nets that kind of move them through the water column. These guys are out there moving and flapping their feet. The Michael Phelps flapping their giant feet of the gastropod world to be predators of other snails in the water column. So they're strong swimmers because of the muscles of the feet and not so much because of the size. And then, of course, if we jump from the mollusks to the annelids, we have some of the classic, most beautiful swimmers, and these are the tomopterids, the polychaetes, that have these gorgeous coordinated parapodia. So you remember the parapodia? So coordinated paddles on either side of the body across the segments, the serial segments from anterior to posterior, that swim them through. And that flexible body plan, the segmentation permitting that, and then the coordination of the body waves, and the giant, if you remember some of the polychaete time we had in 2700, the giant kind of central axon, the nerve axon that permits the fast transmission of these signals, and the coordination across all of these parapods allows that undulation and allows this kind of fantastic movement through the water column. So all sorts of different, so we've got jet propulsion, we've got flapping, we've got paddle swimming and kind of rowing with your feet or your parapods or your mantle or the bell of you if you're a medusoid cnidarian. So all different, like an enormous number of strategies. And I don't see any questions in the chat. We'll keep going, thinking about movement in the marine system. We'll think about, we mentioned echinoderms already in thinking about sea cucumbers and how they're using their kind of tube feet. I mentioned in passing, but also that they can undulate and flap. There's other non-traditional swimmers, non-swimmers that we don't traditionally think of. Often when we think of crinoids, this kind of dominant lineage of echinoderms in this area, when the Silurian Sea that forms the rock that we stand on now was an active and warm ocean floor, it was filled with crinoids. And there are still crinoids in the world, and you're looking at an oddly moving one here that's gone through some kind of a change and some kind of a stress, because the crinoids are the stalked echinoderm, right? So my elbow kind of sitting in the substrate, and then secondary radial symmetry that is allowing kind of these feeding tentacles, almost looking like a little flower, sea stars or sea lilies, that common name referring to their gross morphological appearance. So sessile, but those arms can actually be moved to walk. So the crinoid, the sea lily, can walk, and the sea feather stars like this, they can get up and not just kind of walk, but they can undulate and swim their arms in a coordinated fashion and swim through the water. The classic, or the most iconic of the echinoderms, though, is, and for many of you, if I remember correctly, amongst your favorite invertebrates are these sea stars and the tube feet and the water vascular system that we spent a lot of time talking about in detail last year in 2700. So we're going to mention some of those and mention departures from the crawling that they do, the iconic movement of the tube feet. But it's kind of an incredible adaptation, right? This movement via hydrostatic pressure. So they have the water vascular system, right? Where you have the madreporite. So this is a sea star diagram from one of the texts associated with the course, where it's connected to the outside external environment by this sieve plate, or the madreporite, that brings in the fluid of the ocean and uses it within this water vascular system and distributes it amongst each of the arms out by these long radial canals that are bracketed by these tube feet, the ampulla, kind of the bulb, and the tube foot, terminated with the sucker. And so shortening and contracting the longitudinal muscles of those tube feet and using adhesion, which we won't talk about too much today, allows them to push water from the water vascular system out into the extent of the tube foot and let it, and of course, they're also, if you remember from 2700, there's the tissue. They have the amazing property of this mutably connective tissue. They can harden what is otherwise a soft tissue just with a tap. So the physical action, so water moves from the ampulla out into the tube foot. The tube foot touches the ground and can become harder as it's moving across the ground, and is used in this coordinated fashion to crawl the sea star across. And so most of the motion that you've seen of sea stars is really, this is now moving from across my screen from left to right, the stepping of a single tube foot. The retractor muscles aren't shown, but you can kind of see the compression of the ampulla that sends the liquid out, and the use of the hydrostatic skeleton to move this echinoderm through the water. Now, one of the cool things that we've learned recently about how sea stars move is we've quantified observations that have been made through time about sea stars, and that is that they can hurry. There's slow crawling motion, but there's the sea star that's moving across the bottom. It's galloping. And the thought is that these fast speeds, these galloping sea stars, might be just as the same way that you would use mantle movement for just kind of a squid for kind of sitting in the water column, but if you were going to get away, you were going to like jet propulse using your siphon. These would be decisions involving potential predation or prey capture. Now, what I'd like you to do is to find your oh, sorry, Olivia, I see the question. Two modes that polychaetes, undulation and paddling. Yeah, and the polychaetes that we're talking about principally with the paddling of the parapodia. So a delightfully alliterative answer. The preponderance of paddling parapodia in particular with the annelids. So find your annotation tool, and I'm going to ask you to interpret a galloping sea star. So what I'd like you to do is to draw kind of from time 0 over here. So from 0 through roughly a minute. What horizontal speed, so higher, so you can see there's a slow speeds. This is a sped up video. But what would the crawling speed look like versus the galloping speed? Take a second to think about that, and then find me your annotation tools and give me your impression. This is great. That's awesome. I like this. I like a couple things here. I like that there's, amongst the lines that have appeared already, there's the idea that there's either there is a change clearly from the fast to the crawl. This is why it's a selected trait, that it is getting you there faster. But then you're thinking about whether or not that transition is regimented or gradual. Now, in fact, take a look at the video and try and trace that movement, because there is a fast thrust that is then followed by a refraction period. And if I, without taking yours away, this is actually what the measured speed averaged across a bunch of different runs and averaged across a bunch of times. This is, you've got essentially through crawling to being the galloping, the change that you identified. So from slow to faster. But the key part is this change, this saltatory change or abrupt change is repeated. And so the gallop towards the prey item or away from the predator is continued. And so you've got an injection of speed with a recovery as the tube feet recover and thrust again. So a more consistent slow crawl then to a faster stage that has the change, periodic change in speed. Galloping sea stars. Oh my. Awesome. So there's Caramel's appearance again. We're going to transition now to the terrestrial environment, and we're going to do it with one of the first lineages that did it. First one of the arthropod or the arthropod kin lineages, not an arthropod, an onychophoran. It's its own phylum. These are, you remember Hallucigenia from 2700? So some of the first animals that probably moved from and a lineage that extends back to the Cambrian explosion, but one of the first ones that came out with their tarsal claws and these lobe feet. These lobe feet are a hydroskeleton in the same way. This is kind of the link back to the echinoderms. This is not because of a shared relationship. It's just that in this kind of moving animal that molts, right? This is the same molting animal that has these feet tipped with their little tarsal claws, but they're moving terrestrially via unjointed limbs called lopopods that they inherited from Hallucigenia, and these lopopods are moved hydrostatically. So that's the key. That's the common operational or mechanistic link to the echinoderms. And what the lopopods, what's happening is that as this foot moves, it's being levered by muscles in the body wall that kind of take it and depress it and forward, and then we've got it backwards, and then so we've got kind of a paddling movement of these soft extensions of the body wall. And what's happening is that there is, in the walking direction pictured on the top, the onychophoran on the top is going off to the left-hand side, And so we've there in the video is now slowed down to these individual pictures, but we've got the depression of the claw, but then the lifting of the claw and the lifting of the lopopod itself when the intrinsic muscles in the body wall and within the leg are activated. This is kind of amazing. This is so very similar, perhaps verging towards convergent kind of a strategy of lopopod, of hydrostatic legs. So I said it's kind of convergent because these are not tube feet. There's no adhesion, but they are legs that are coordinated through a hydrostatic skeleton and not through extensor and hard exoskeletons like we will see eventually with the arthropods. Now, larval arthropods, which we're going to jump to in a second, lots of the onychophorans, historically, like deep history-wise, people thought they might have been caterpillars. They thought there was a lot of similarity between larval Lepidoptera or larval insects that move ostensibly in a very similar way. So we have a soft body with legs through abdominal segments and then some thoracic segments that have more cutaneous kind of claws. But then these soft abdominal legs and the terminal proleg right back here, historically this was thought to be done also with small muscles in the body wall. And what we're seeing, again, as we can use more and more high-resolution camera footage that we can deploy in more and more naturally or near natural environments and take videos at higher and higher speeds and higher rates. These advanced imaging techniques that we talked about with respect to flying as being a real key change in our providing kind of thrust to our changing understanding of how flight is achieved across different lineages. Here, it's changed how we understand caterpillars moving. And this led to this paper that was produced a couple of years ago, Visceral Locomotory Pistoning of Crawling Caterpillars. So the movement, the wave-like, so instead of looking at the legs as this kind of caterpillar moves across, look at the wave, the undulation, that kind of boom, that undulation, that wave-like motion on the dorsal side of the body. So that's driving the movement of the legs, or at least in this video it appears coincident. But this is actually a wave that's proceeding across the whole body that is bringing the legs along with it, but it's actually being driven by muscles and by the gut. And remember, the gut of a caterpillar, because it's eating plants, is a long, complicated thing. It takes up a lot of the body, and often it's very full. And so it's being used as a mechanical piston here to move the body and to push it forward. So the viscera, the body wall, the middle section, doesn't move so much through this video, so from top, or video frames from top to bottom, but the position of the gut and the positions of those segments in the gut drive that forward, and so are driving the body wall, the body forward essentially via gut undulations, and the legs are being kind of like following along backwards. These are particularly abdominal legs. But that's kind of an amazing. So in addition to thinking about analogous systems, analogous human systems, we have propulsion, jet propulsion that we talked about with the flying moths, the death hawk moth on Monday, and propulsion we talked about more again today with squids and with cnidarians. And now we've brought in pistons, and we're thinking about pistons instead of being the driving piston of an engine. We're thinking about it being the driving gut that flops the legs forward and into place. Invertebrates, all of these things they've come up with first, right? Like, we're so late on the scene. It just we should really be paying more attention to who is who amongst these species that share their planet with us, because there are so many solutions that we would benefit from. And we'll talk about that some in the next section when we talk about walking and running and move to a terrestrial setting, largely for all of this. So if you are in all of these images here, so we've got leafcutter ants and pseudomyrmex ants and army ants and Phydoly ants all in northwestern Costa Rica. The number, there's kind of across, so this is kind of one lineage within the hexapods, but there's three independent origins of legged locomotion in the arthropods. That we see it in the non-monophyletic crustacean. We see it in the chelicerates, so spiders and their relatives, and we see it in the hexapods, the insects and their relatives. And things vary across all of those in terms of the number of appendages, because that varies quite a bit. This is, of course, the videos are classic 3 legs. The costs associated with legged locomotion have to do with specialization. The benefits are that you can move really well, and this is one of the things that we're going to talk about today. That they can also be used, legs can be used for other things. They can be used for handling prey while you move, or they can be used for reproduction also. And so there is a cost associated with specialization, but there are benefits that just based on the preponderance in the record of the taxa that live like this, that suggests the costs outweigh the benefits. So if you think back again to 2700, and we think about the time that we spent talking about arthropods, and we talked about the importance of segmentation in a lot amongst the phyla of the world, the phyla of the invertebrate phyla of the world. We said it was really true segmentation we see most iconically within the annelids. And when we talked about arthropods, they're still segmented. Like we see the 3 tagmata, like within the insects, we've got a head and a thorax and an abdomen. And we didn't talk about the legs being segmented because that's not anterior to posterior. We talked about them being articulated. And so these articulations, which are what the yellow lines are drawing attention to here, is that there, amongst the 6 walking legs, there's 18 total joints that must be manipulated to walk. That's potentially expensive from a neurotransmittery perspective, but it drives the number of possible movements in space way up and provides many, many different solutions to stability and to being stable while moving and predatory or escaping, but also stable while resting. The static stability that you're seeing here. So we've got articulations. So bring those arrows down here. And you look at this kind of, so we're looking, if we wanted to draw 2 eyes right there looking at us, this is a kind of a cross-section of a hexapod, of an arthropod, of an insect, looking at you while statically stable, hung, suspended, and supporting the insect off the ground, giving it a low center of gravity, which is very stable, and in fact, an unusually stable kind of way to exist in the world. Now, articulated legs are dependent upon, as we know them from the biological record, are dependent upon articles or hard parts of the appendage. So cuticle, right? So there's the little text reminder, don't call them segments. But the muscles that drive this are inside of, so this is not a hydrostatic skeleton, right? These are antagonistic pairs of muscles that work across. We've got flexors and extensors across each one of those articulations that permit really sensitive, stable stability just passively, but then also really sensitive responses to the environment that allow them to move through environments from the aquatic to the subterranean with the simplicity of the terrestrial in between. So there's a reminder of where your kind of extensor and flexor muscles are inside and across that one of those articulations. And if we zoom in, you can see the flexor and extensor again, kind of more close up in this diagram from your Bresca text, where the direction there, this is, if you want to think of this anthropomorphically as a knee, as it bends over there, we're seeing the flexor muscle contract and pull across this articulating membrane. And this results in a different, so very stable, passively stable way of existing, but also great stability while it's moving. So this is some vertebrate examples, like 19 lectures in, and we're going to show you a vertebrate example of things that you know. And this is looking at kind of the gait patterns for a 4-legged or a bipedal organism like ourselves. So we go right, left, right, left, right, left, right, correct? So that the rectangles there, the colors indicate, the colors are associated with one of the appendages, and then their appearance is when it's in contact with the ground. And so we've got then a diagonal gait that where we see the left, the left rear, and so that kind of bounding action of the tiny mouse. But so those are bipedal and diagonal motions. But then the tripod motion of the three-legged insect is an extremely, so L2, R1, and R3. So L2, R1, and R3. So this makes kind of the tripod that we were talking about. So L2, R1, and R3. And so this is, so we go through there with one step, and then we have the second triangle that supports. So we've got basically triangles that are oscillating across. And what we've, didn't actually accomplish what I wanted to do. So that triangle, that tripod gait, middle one side, anterior, posterior on the other, and this provides a real stable support during walking. So we've got there the two different triangles. And this is a video. You can see slight kind of bobbing of that fly as it's moving, using its tripod as the fly is walking. But you can see, if you track through the L2 timing with the R3 and the R1, moving that tripod stably through its environment. The stability indicated just by the very nearly straight line. Now insects have used this tripod, so the articulations of the legs and the tripod of their gait, great stability while not moving or while moving, to their advantage in a number of different environments. And one of the examples that we'll talk about today is with ants. This is a study that you might have heard about. It's several years old now. But it involved this cataglyphus ant that navigate, or apparently seem to navigate, a vast desert habitat by what was called path integration. And so they're continuously integrating directions. It wasn't entirely clear what kind of directions they were integrating. Were they looking at celestial nighttime orientation clues from the sky? But how were they? And so the answer is partially yes to that, but that's not the part we're going to unpack today. Because the directionality that the stars could provide is really only useful if they can count the direction, or if they can enumerate the distance that they've traveled. And so the question in this particular elegant or brutal cataglyph study was, how are they enumerating the distances that they've traveled? And one hypothesis was step counting, that they were counting those triangular gaits and saying the higher the number, the longer the distance. How did the scientists test this hypothesis? Well, this is the brutal part. They took an experimental group in the middle where they did nothing to, and they just counted them. And then so their legs were the same length. And then they made two experimental treatments where they cut off the legs, they made them shorter, or they added a section with glue. And this so in this group, the ants on the stilts were overestimating their travel distance, and here with the stumps, they were underestimating it. And the way this manifested itself in the experiment was they would take them away from their colony once their colony had been kind of acclimated to an experimental arena. They were moved from the colony entrance, and then the number of steps they took to get back was known. And so the control, they just kind of walked, say, was 11 steps. The stumped treatment would walk 11 steps back and then do characteristic behavior to search for the colony entrance, but they were meters shy or centimeters shy of the entrance. So even though they could see it, they began, in spite of that, they did their step counting, and they were like, it's got to be here, I've gone my 7 steps. With the stilts, they overshot the colony entrance and then started to did the characteristic look around for your colony entrance. So it appears this desert ant is using not just the stars to get a direction, but it's step counting using its legs to figure out its distance. And here is a robot, the Antbot, that was designed for autonomous return to a release point, potentially useful in farming strategies in the future with automation. But this Antbot was designed and deployed using the same strategies that are employed by the cataglyph ant. Now, we talked about independent derivation of segmented of walking legs. The myriapods, the millipedes and the centipedes. So this is one or two. This is a millipede, and you can see two segments, two legs or parent legs for every segment or diplo segment. And that undulation, that metachronal wave that kind of moves up and down the body, these wave-like patterns as they propel themselves via one, in the case of the centipedes, or two segment pairs of legs per segment in the millipede. And here's another millipede from Costa Rica, but the diagram itself is kind of showing you the forces that are involved at the tips of the legs for, in this case, a centipede as it moves through a slightly viscous material that they've videoed afterwards. And you can see, if you watch that, the movement direction is pretty consistently from left to right, but the coordinated kind of motion of the legs on each on left and right side, but then from anterior and posterior of the body. Now, going back to the insects for a second, one of the most iconic ones, the ones that we're told to expect to survive us, most of them will outlive our own species, but the cockroach. This moves with, its gait is very straight on. It doesn't undulate. It doesn't move itself or bound like the, as we saw kind of iconically today with the echinoderm. But it has a ballistic phase because it has a tarsus and a pretarsus that are clawed that allow them to climb, and its antennae are using it to guide its walking. But as it's moving around, they can compress their exoskeletons. So from the lateral to ventral compression that allows them to move through, in this slowed down video here, a crevice height of 3 millimeters. So they're compressing their exoskeleton, and their mobility isn't really impinged because the articulation of the legs allows them just to kind of like fold through afterwards. But their forces that they experience, that they're quite adept at dealing with, are estimated to be 300 times a body weight as they get through that tiny, tiny crevice. Now, one of the final examples, we talked about bipedal locomotion, or we didn't talk about it, we mentioned it and used us as a left-right example early on. We could change that iconography and think about the bipedal locomotion with mollusks. Now, historically, we used to think that this was required. It couldn't be done unless you had muscles against a rigid skeleton. But 20 years ago, there was a science paper that reported two species of octopus, in Indonesia, one of them's being shown here, that used two of its legs to walk. And so this has been filmed many other times since then. It's kind of as you can imagine, if you were diving and you came across this, why you would want to be filming this. But instead of using muscles across a hard skeleton like we do, it's using transverse, so kind of muscles, antagonistic muscles, in the same way that we would see undulating worms move, but they're using them so transverse and oblique bands of muscles in the arms that give them the exceptional flexibility, and then it's a hydrostatic skeleton again that is allowing the support and the walking of these two legs. And just as we saw with the ants, engineers have been inspired to create robots, an octopus-inspired bipedal walking robot that can go forward. Here it is kind of moving through water, running kind of gleefully there through the water. And this is what it looks like. So not perhaps a robot that you might see in Alien Earth or the worst season ever of Battlestar Galactica, but another example of an engineering solution, a low-energy engineering solution that we've applied based on inspirations gathered from the invertebrate world. All right. This is about all of the time that we have today. So, swimming, crawling, walking, and running. Just a quick touchdown in each one of these categories across the invertebrate world with some examples. Where are we going? Well, next week we're going to be spending time in the lectures thinking about diversity. We're going to talk about the number of species, how we estimate the number of species, which is kind of a thing that we ought to be thinking about with the invertebrates, because it's really the number of species is an invertebrate question. Next week's labs, is a work period for your presentations. Check CourseLink for any kind of updates from Shayla on that. And then don't forget that your final draft is due Friday for your lab report. Work along on your presentations. And as we approach the end of the term, kind of roughly a month from today is when your final exam is. So these details will be in the PDF associated with the lecture today. But the lecture, there's no surprises in the lecture. The lecture is like all of the case studies along the way. The content is from the content lectures from 2 to 22, and there'll be multi-state questions, roughly 3 of them from every lecture. So there'll be about 60 questions. But the context is the same, that it's designed with universal design principles in mind. So there's no lockdown anything. That it's open on the 10th of December. Even if your web advisor tells you it's from a specific time, you can write it any time within that 24-hour period. And it's a 2-hour exam, but you have everyone has 2 times the time to write it. So I'm going to stop here. I'm going to stop recording. And if you've got any questions about any of this, hang around, ask them to me. Otherwise, take care of yourselves, take care of those around you, and we will see you next week.